So what are the things that changes the factors affecting the expression of the regulatory genes? So these are, remember, the regulatory genes are the type of genes that turn the genes on and off. So yeah, turn the whole thing on and off, basically. So firstly, we've got the change in temperature. So this is for both plant, eukaryotic and prokaryotic, whereas temperature, it can be internal and external in eukaryotic, but only external in prokaryotic, single cellular. I can't see that, but that is sunlight, the intensity of sunlight. That isn't specifically for humans, that's for eukaryotes such as plants and etc, those sort of cells. And internal factors such as, once again this is for eukaryotes, such as hormones and psychological stress. So this changes the expression of the hormones that can be involved in the expression of the regulatory genes. And obviously the pill formaldehyde and drugs can change this. Transcription factors that impact the genes. So transcription, obviously copying the single DNA strand with the mRNA, with the DNA polymerase. Those transcription factors that impact the genes include, so the transcription factors, these transcription factors include, number 1, short gestation. No, that's not transcription factors also impact genes. So transcription factors impact the polarity of the embryo. Which means what side is the head and the tail. So, it just depends on what side, the cap and the tail is on. And also, so that's basically what the transcription factors is. But flies, let's put that there. Flies are really often used in genetic experiments. So, just do some. That's a fly. So why would a fly be good for testing and to use genetic information for? So they're easy to study homobox genes because they have a short gestation period. So that means they reproduce fast, they grow up quickly, you don't have to wait years like you would with a human. Short gestation, so they reproduce quite fast. They have a simpler or smaller genome. Short genome. That's why we also look at the lac operon quite a lot, because it's quite a small genome, while humans have a lot of a long complex genome. Flies only have short genomes, like a bacterium does. You can have a lot of flies in a small space. There's not tight ethics or ethical controls on flies. There's not a lot of ethical control. And finally, it's actually quite cheap. Flies aren't expensive to keep in a container that don't eat much. It's just cheap to actually manufacture the experiment. So if we just go on quickly to a bit of Alzheimer's. So, Alzheimer's disease. Alzheimer's disease impacts a lot of people. Basically, within the DNA, after the transcription, producing exons with a cap and a tail, there is splicing, which takes away the introns from the RNA to produce mature RNA or mRNA. However, Alzheimer's is to do with the splicing, which takes out the introns. There's a failure in splicing, so it's a splicing failure. There's a problem with the splicing process. This results in a non-functional protein. As the exons code for are genes that codes for proteins, this splicing failure causes proteins to be non-functional. proteins non-functional. So the proteins aren't able to be used. This therefore causes plaques of proteins, or they're called plaques, to be formed. These plaques obviously cause problem. Just like a plaque in the artery, they block up some of the space that should be used to transport things, and these non-functional proteins of plaques interfere with neuronal communication, so communications in the brain. The often symptoms are clearly memory loss, most prominent one, but also imbalance, so people losing their stability. Imbalance. But these plaques are easy to see under a low powered microscope. The estrogen is a just put that there. Estrogen is a hormone, and there's an effect of estrogen on gene transcription. So, if I just go through the steps that would the estrogen interfere with the gene transcription. So firstly, the hormone estrogen, for example, binds to the surface of the target cell. So that's the cell. I'm going to put an O for estrogen, which it funnily enough starts with. So it actually binds to the cell surface. Don't know if that's big enough. Of the target cell. Secondly, the hormone receptor complex, that's the cell. After the estrogen binds, there is a hormone receptor complex. So the hormone is the estrogen, the receptor is the receptor on the cell, and that makes a hormone receptor complex. And this activates enzymes inside the cell. So without the hormone actually getting into the cell, it just activates the enzymes, which are yellow dots, inside the cell. This results in a production of the chemical second messengers. So second messengers inside the cell. For an example, CAMP is a second messenger only inside the cell, which are activated by the enzymes from the hormone receptor complex, which is on the outside of the cell membrane. The second messenger causes a series of chemical changes to produce the required response. So the second messenger just changes whatever it needs to through chemical reactions to produce whatever is needed from the hormone that's desired. Insulin, for example, it regulates the blood sugar when you don't have enough sugar. So it regulates it, and it converts glucose into glycogen. So insulin is very renowned, and it's well known for glucose into glycogen. This is in liver most of the time, and also in muscles for short, quick energy responses. Lipid hormones can diffuse easily through membranes. So most of the hormones are lipid, so they can diffuse straight through the membrane. Estrogen is not lipid, so it can't diffuse, so it can't pass through the membrane. But once the lipid is inside the cell, it binds, or is inside the cell, the estrogen binds on the site of the transcription factor molecule that complements it. So it can only bind to the receptor that complements it, otherwise it won't even bind, and therefore if you have too many hormones, it won't even bind, so it won't be enough to be filled up. On binding, the estrogen causes the transcription factor to take place. So this releases the inhibitor molecule from the DNA, from the transcription factor, and this only works with cell membrane on complementary protein shape. So it has to be complementary to the cell membrane has to be complementary to the protein shape, or it won't activate any second messengers or any hormone responses, because it won't even fit to the active site.